Good morning, class 8. Today we are going to see the new lesson, microorganism, friend and foe. Before that, I just want to clarify your doubts in the previous lesson, crop management. This is the concept map of the crop management, production and crop management. Is there any doubt you can ask me? So I will explain the gist of it. So under the crop production and management, first we need to cultivate the purpose of the crop production and management is to cultivate plants. That means cultivation of plants and raising of animals for human use. Together we call it as agriculture. Okay, so when coming to the agriculture, growing of plants and also it includes the fruits, vegetables, all those things. So when you want to grow a plant, we need to prepare the soil. In order to prepare the soil, we need to plow it, and after the plowing, the loosening and turning of the soil happens. So we need to level it. So next to plowing is leveling. Then the leveling also sometimes in some cases includes manure. So where the soil are not fertile, so we need to manure it during the leveling stage itself, because we are turning the soil, right? So plowing, leveling includes manure also. And the next step is the sowing. And sowing, we are having different methods. In that, using the hand, we are sowing the seeds, that is broadcasting, and using the seed drill, we are sowing the seeds, that is the mechanical method. I mean the tradition, not the tradition, modern method. So that is the seed drill. And in few cases, like the plants and vegetables like tomatoes, chilies, and cereal plants, mostly we do transplantation, that is growing the seedlings, plant saplings separately, and again we are taking it and planting it in a rows, like in the rice field you might have seen. So this is the second step. So after this, we need to harvest. The grown crops are to be harvested. So in between, only between the harvesting and after sowing the seeds, we have two more steps, that is weeding and pest control. So controlling the pests like rat, fungi, insects is very important to protect the crops. So pesticides are used. Weedicide is nothing but removing the unwanted plant which grow along with the plant. So these two steps we include after the transplantation or seed sowing, that we term it as crop protection. After this, the seeds has been grown well and the plants are ready for harvesting. So the mature crops are get harvested, that is harvesting. So once we get we have harvested the plants, after that we will be going with the threshing, separation of grains from crops, and winnowing, separation of chaff from grains. So after all this, what is the next step? We need to store this. So storing of grains and also to prevent the growth of fungal, these steps and all taken during the storage. So these are the steps involved in the cultivation of crops. I hope you all understood. Is there any doubt? Okay, the next one, a few points to be added between the sowing and harvesting, that is maintaining of the nutrition. So as I said, manure is added in the first step itself. We need to undergo the nutrition management of the soil after preparing the soil to ensure that the enough of nutrition is available for the plants. Okay, so we have few methods, natural methods, that is feed fallow, leaving the land for few months and years without cultivating anything, and then we will use it, so that will regain the soil fertility. Then crop rotation of crops, first seasons legume plants, second season non-legume plants, so thereby the soil fertility is maintained. And next one is the mixed cropping, together with the legume plants we can grow the other plants, so thereby there is a circulation of nutrients. These are the few methods in natural methods. And you all know the artificial method is fertilizer, usage of fertilizer, urea, NPK, and specific fertilizers. Phosphate means you can use super phosphate, potassium you need means you can use potash. So potash is the potassium rich fertilizer, and phosphate, super phosphate. Okay, and urea contains both the nitrogen, phosphorus and potassium, that means NPK. Okay, most widely used fertilizer is NPK and urea. So these are the points you need to remember. Then coming to the irrigation, after sowing the seeds, we irrigate the seeds. So the traditional method includes mode, chain pump, rahat and dekli, and modern methods include drip irrigation and sprinkler. So these are the few things we need to add. I hope you are clear with the concepts and the steps involved in crop production and management. Shall we move on to the next lesson, that is microorganism, friend and foe. So microorganisms are microorganisms which you cannot see with your naked eyes. With the help of the microscope only you can see. So they have a different structure to protect themselves from the environment. So that outer hard covering we call it as cyst around themselves. For example, this is the cell. So around the cell there is a one layer of outer covering like this. So that is termed as cyst. So they do this when conditions are unfavorable. So for example, they are living in a place where they need a nutrition, the nutrition is not available. For anaerobic bacteria, anaerobic means they doesn't need oxygen. Okay, they need some other kind of gases like carbon dioxide, sulfur or any kind of gases they need. So they are anaerobic bacteria. Sulfur bacteria and all we have. So they need sulfur like that. So they are anaerobic bacteria. They live only in the anaerobic condition in the absence of oxygen. Likewise, aerobic bacteria we have. So that is, the aerobic bacteria are nothing but those bacteria which live only in the presence of oxygen. Okay, so if the oxygen is absent now, so they cannot live in the condition, that is called as unfavorable condition. So this unfavorable condition lead to the formation of the cyst. Okay, they survive by remaining inactive within the cyst. Okay, they won't reproduce, they won't active, they won't cause any diseases. So they want to protect their cell content, they remain inactive inside the cyst. Okay, so this kind of microorganisms you can find in everywhere, air, water, soil, even on the rocks, everywhere you can, mountain, hill station, plains, everywhere you can find the microorganisms. So some of the microorganisms live in group, some will live in colonies. I mean some will live alone, separate microorganisms also there. Some will live in groups like this. Okay. So coming to the discovery of the microorganisms, the person who was the first one who to discover the microorganism, that is the bacteria, is Antony von Leeuwenhoek. So he has used a screwed microscope to view this microorganisms, and he has found out some smaller structures which are living. Okay, and Robert took cell that is in honeycomb structure. This structure, it is a dead cork cell. He has seen this structure, and later only it was considered that the cell is a compartment, small compartment, like that they have uttered later. Okay, and coming to the different kinds of microorganisms, we have different kinds of microorganisms. Usually we term autotrophs for the organisms which will prepare their own food. Even in bacteria we have autotrophs like algae, which contain chlorophyll. Those organisms prepare their own food, they won't depend on other organisms. So thereby there is no parasitic kind found in this kind of organism. So now coming to the bacteria. So singular form of bacteria is bacterium as given here, and for fungi it is fungus, and for protozoa it is protozoan, and for algae it is alga, and viruses it is virus. So these are the singular and the plural forms in the names. all are single cellular microorganisms except fungi. The only fungi which is single cellular organism is yeast. Okay, so these are the things we need to know before entering into this chapter. So they have cell wall. The cell wall is present means yes, of course the cell wall is present, but the thing is they doesn't contain a definite nucleus. See, the nucleus are randomly present in the cytoplasm. So this is the cell wall and the empty spaces, no? So this spaces we term it as the cytoplasm. It is fully like this, okay, cytoplasm, and the genetic material is scattered here and there because of the absence of nucleus. Nucleus means what? Nucleus will be the one which contain 3 layers. So in our cell we have 3 parts in the nucleus. So this is the cell. So we have 3 parts, and this is the outer membrane called nuclear membrane. Okay, and inside nuclear membrane we have nucleoplasm. Inside nuclear membrane we have nucleoplasm. Okay, we have nucleoplasm, and inside that we have the production unit, nucleolus. Nucleoplasm, nucleolus, which is the production unit for the ribosome. And in this nuclear membrane, there are small pores. Pores means what? Holes. So that holes will be used for releasing the ribosomes into the cytoplasm. Okay, this is how the ribosomes are manufactured and released. So there are 3 parts in a perfect nucleus, that is definite nucleus. Nuclear membrane, this one is nucleolus. Okay, and it is the cytoplasm of the nucleus is called as nucleoplasm. So these are the important parts. If these 3 parts are not present, the nuclear membrane is absent, it is termed as nucleoid. So this kind of nucleus only is present in the bacteria. So the bacteria doesn't have perfect nucleus, so it is termed as nucleoid. So this is the main difference between the bacterial cell and the other single celled organism. Okay, so we term it as prokaryote. This kind of nucleus present single cell is known as prokaryotes. As I said before, the perfect nucleus, no. If the single cell have a perfect nucleus with all those parts I have said, no, so those are coming under eukaryotes. These 2 things you need to remember. Okay, any doubt in this? Okay, now we will see about the microorganisms. So microorganisms, as I said, the microscope, as I said, the microscope is the one which is helpful for viewing the microorganisms. We have 2 kinds of microscope, that is light microscope and electron microscope. So these are very 2 different microscope. Light microscope is a very simple microscope. Using the light, the specimen is nothing but, for example, this is the slide. Inside there, whatever the specimen you want, for example, you are taking any tissue or anything, any slice of leaf or a small microorganism, fungus which you seen in the bread. So you can take it and keep it in center of the slide, a small glass slide, and fix it. Okay, so then it is termed as the specimen, whatever the specimen may be. It is viewed under the microscope lens. So you can see only the outer structure like cell wall, shape of the cell. So that only you can see through the light microscope. But electron microscopes are very complex, and you can see the interior structure of the cell, like the nucleus of the cell, mitochondria, Golgi apparatus, whatever the cell organ is present inside the cell, that can be viewed through the electron microscope. This is the difference between the electron microscope and the light microscope. In electron microscope, beams of electron are used instead of the light to provide the maximum of magnification. Magnification means that zooming. How much you can magnify in light microscope? Only 25 to 400 x. Okay, you can see. But in electron microscope you can see even more than this, you can magnify. So magnetic fields are used to bend the electron beams used in the electron microscope, like the rays of the light. Okay, and thus it helps us to view the internal structure of the cell. So far we have seen about the microscope, different kinds of microorganism and its structure, what are the difference between the prokaryotes and the eukaryotes, right? And we have seen the microorganisms, where all it is present, how it will be inactive. So this is the introduction about the microorganism. So in next class we will see about the bacterial shape, size, habitat, cell structure, nutrition and reproduction. Sir, any doubt? Thank you, children. Bye.